Good? Okay, so we have two phage. We have ToneTone and A6 lysogenic phage, and sprinklers SE1 lytic phage. ToneTone has 3 tRNAs clustered at the beginning of the genome. ToneTone has 30 tRNAs, 1 tmRNA, and peptidyl tRNA hydrolase. Sprinklers has the majority of its tRNAs clustered in the second half of the genome.
Figure 2 shows factors that either increase or decrease the pool of tRNAs in a cell. You can find the ones we found in sprinklers boxed in green. If too many peptidyl tRNAs accumulate in the cell, protein synthesis can no longer occur. And what happens is the peptidyl tRNAs are released from the ribosome, and then peptidyl tRNA hydrolase comes in and cleaves the bond, which is found in panel B of the 3. And tmRNA does not increase the release of peptidyl tRNA from the ribosome, but it still facilitates in the cleavage of the peptide from the tRNA. And overall, this will increase the pool of tRNAs available for the cell. You can note that release factor 3 is found in figure 2. However, we did not find that release factor in sprinklers or ToneTone, but we did find the release factor 1 in sprinklers. And the function of a release factor is shown in figure 3, panel A. The release factor binds to the stop codon found in the A site, and then this facilitates the release of the polypeptide.
So if you look at figure 4, we see one system that helps recycle stalled ribosomes. We see the tmRNA small protein B complex. It comes into a stalled ribosome. It transfers the messed up protein to the complex, releasing the tRNA that was stuck in the ribosome. That messed up protein is then tagged for degradation and released. At the same time, a release factor is recruited by the complex, and that release factor allows the ribosome to be recycled back into its two component parts.
Figure 5 was comparing the two types of disparity in codon usage frequencies. We use this through Kazuza, which is a codon database, which outputs codon frequency. And we collected data for ToneTone, sprinklers, and Mycobacterium smegmatis. ToneTone only contains 1 tRNA that reads a codon for asparagine and glutamine. However, sprinklers contains 1 tRNA that reads a codon for asparagine and 2 codons, or 2 tRNAs that read a codon for glutamine. And what we found was sprinklers and ToneTone and Mycobacterium smegmatis shared the same codon frequency bias. And this is important in conserved codons because their genes are readily expressed, and so this is necessary if phage has a certain function, so we can make sure the protein gets translated efficiently.
And if you look at figure 6, we gathered the data using the same program, Kazuza. But here we look at stop codons. Each stop codon is recognized by a specific release factor. Here we see those for release factor 1, 2, and 3. ToneTone has no release factors. Sprinklers contains release factor 1, so we anticipated that there would be a bias toward the stop codon recognized by release factor 1 for sprinklers. This is not the case. Both phage show the stop codon preference for release factor 2. This is the same as our host, Mycobacterium smegmatis. Therefore, we suspect that the phage are using the release factor 2 produced by Mycobacterium smegmatis.
And something notable about this figure is you can see release factor 3 is not preferred that much, and that could be because in this figure it's showing that release factor 3 actually increases the amount of peptidyl tRNAs, and like I mentioned before, this is not favorable for protein synthesis.
In the future, we would like to look and see if perhaps in sprinklers somewhere in the unrecognized genes there is a release factor 2. However, we don't think this is highly possible. We suspect instead that sprinklers and ToneTone both use the release factor produced by unsmeg.
Also in the future, we would like to determine if tmRNA and peptidyl hydrolase are beneficial to sprinklers, and we can do this by deleting those genes. And then we can compare sprinklers with the genes deleted and sprinklers with the genes and see if there is a difference in lysis rate. If the lysis rate decreases with the deleted genes, that means these genes probably are beneficial to sprinklers. However, if the lysis rate does not show a change, we can assume that these genes were presented through horizontal exchange, and it just requires more time for them to lose these genes throughout their genome.
So all of what we looked at are factors that can increase the tRNA pool in the cell, which helps with protein fixation.